And so, let's see here, somehow I got, there we go. Like I said, his work, especially at the second half of his career, was really important for linking the fields of evolutionary and microbiology. And I was really myself influenced by a set of 6 articles in molecular evolution called Molecular Evolution in Genetics, sorry, called Molecular Evolution of the E. coli chromosome. They used to do these series where they'd have molecular evolution of E. coli chromosome 1, 2, 3, etc. And there was a series of 6 articles, which you should check out. The one that is kind of cited the most often that made a big impression on me, and I think many others, was this one about clonal frames, where he sets out this idea of bacteria having a clonal frame in their chromosome that is punctuated by other components that are recombining or coming in elements from other places. As you know, that has now turned into the kind of this pan-genome concept, where you have this vertical transmission through single mutations of the clonal frame, and then other pieces of DNA coming in through homologous recombination, non-homologous recombination, which is a really important innovation in understanding the evolutionary dynamics, molecular evolution of bacteria that are reproduced asexually, but still have gene flow happening through other ways. Okay, and he was really actually well known for being a very good teacher and speaker. He used music and poetry and even dance in his lectures. So he had a dance of the chromosomes that he used to look at the way, to show students the way that chromosomes segregate. And I think that Lizzy Wilbanks is kind of the perfect person to be giving this lecture. And I really felt that way when I was watching her give her lecture in the field this week, which was absolutely amazing lecture. You all know because you were there. So here she's doing, I think, the dance of the electrons, bringing the electrons being moving on the electron tower, being oxidized and reduced in sulfate metabolism. So this is very perfect for Lizzy, because that's what she started in chemistry and biology as an undergraduate, and then went to do her PhD at Davis. And while she was there, she came to the Microbial Diversity course, which is where she found her complete inspiration for the rest of her career, which of course you all know were the pink berries that she's been studying in her lab ever since. So she kind of put together her own PhD to look at that from the professors at Davis that were willing to think outside the box and work with Lizzy on this. And she took that to start that project to start her career at her professorship at the University of California, Santa Barbara, where she's an assistant professor in the Department of Ecology, Evolution, and Marine Biology. And she's looking at both metabolic diversity and also the community and population diversity using genomics and metagenomics, which fits, I think, in with the ideas that Milkman really started to introduce. So I'm ready to hand over the mic, if you will, to Lizzy.
Thank you, Rachel, and thank you all of you for having me here today. It's an incredible honor to be here, and I'm just, as you can tell, really, really psyched. I'm so psyched that I pretty much lost my voice. So bear with me croaking through today's lecture. You know, as Rachel said, perhaps generously with professors at Davis that let me think outside the box. I'd say they let me run feral. And I'm so grateful to all the folks for having me back to the course, and for the course for sort of sponsoring my career for all of these years and really helping me find what I was excited about working with and giving me the freedom to do this. Looks like we're having some technical difficulties over here, so I'm just going to start talking. So I came to the course as a second year grad student, and I've been working on a complete bioinformatics project starting off. I had been looking at chromatin immunoprecipitation, trying to find transcription factor binding sites in our killed genomes. And I had never done any programming when I got to grad school. I'd never worked with sequence data. My undergrad research was in synthetic organic chemistry. And so I had really no idea why anyone took me in microbiology grad school starting out. I had no experience and a lot of ideas. And yeah, Scott's like, it wasn't my fault. Yeah, he was on the admissions committee, so I guess it's his fault. But and so it was a rocky start. And a lot of folks take a little time to figure out what they want to do. And I ran straight through following the woman of my dreams who knew exactly what she wanted to do. So I was following my then girlfriend, now wife, to Davis, where she was a veterinarian, and I sort of figured it out as I went. But I came into the Microbial Diversity course at the end of my second year, pretty burnt out with what I was working on and really not sure where I wanted to go next. And what I found here was just a real blessing. And so, I'm going to start off here with we've broken the clicker now. Awesome. All right. So, this tree that you've been looking at of the microbial diversity tree here, and then let's see. Oh, the clicker's not working at all. Okay. So we'll go here. So there's me showing up in 2010, getting really excited about not being in front of a computer anymore, and being out in the field, getting really muddy, and occasionally stuck and having to be rescued and Sipoised by folks who are now my friends and collaborators. And I found this just while we were getting ready on Facebook. Here's a picture from 2010 of me holding a berry in a tube and getting really excited about it. So I've been looking at these for a long time. But I think one of the things that's been the most different and exciting for me about my career has been how many people from this tree, from this course, have worked on these throughout the year. I've gotten in cabs with people going to a conference and found out that they were the course and that they worked on berries in years past. And it's just been an incredibly fun way to connect with a really diverse group of people who also thought these things were weird. And a lot of folks will talk about the work that is being done in my lab and the work that we published, but there were volumes of unpublished things that the work that I've done has built upon, and things that people have told me to try, data that people have sent me. It's an incredible legacy that I feel really, really privileged to get to be a part of, in studying these weird little balls of pink goo that literally glue us together. So I wanted to start off here just acknowledging the current we of the Berry fans, which is my lab group. Most of you have by this point met, at least most of you in this room, have met Braulio and Aaron, who are my graduate students who are here with me doing research. And they're part of the Berry crew, where I'm also going to talk about work today that's been done by my postdoc, Dr. Hugo Doré, who will be joining us here in about 2 weeks. So more Berryites that you'll get to meet. And then also postdoc working with my group and with Michelle O'Malley, Dr. Amy Eisenberg, whose work I'll talk about a little bit today. Our undergrad extraordinaire, Fox Bravo, and then Tori Jones, who's a new master's student in the group, who I'll touch on her project at the very end. I don't have a photo of her. The rest of my group here is actually working on the kelp microbiome, so in search of something local and slimy. This is our new system that we're working on here at Davis, and then of course my funding sources for the work that I'm going to present today, mostly through the Institute for Collaborative Biotech, which is an Army Institute funded at UCSB. So broadly, my group works on microbial biofilms and aggregates and thinking about these in the context of microbial interactions. And this week, our theme has really been building up from simpler single models, population models of microbial communities to the whole ecosystem. And so one of the things I'm going to try to highlight throughout my talk today is ways that we can track the interactions of wild microbes. And someone asked a really great question, why don't we just do this in the ecosystem? And I'm going to show you some of the ways that we're trying to do that. And I think as I show them to you, I'm going to try and highlight some of the challenges that we have with them, right? What are the tools that we're going to use to peer in and watch these microbial interactions? You know, as microbial ecologists, we have a lot of problems just watching the lions interact with the gazelle, right? Even being able to do that is a feat for us, whereas for other ecologists, you just need a good pair of binoculars and a hat, right? So, and this challenge really is kind of summed up by thinking about the incredible scale that microbes sort of occur over, right? From single cells up to ecosystems spanning nanometers to kilometers. And to do this, our tools really need to span an impressive array of resolution to throughput, right? If we want to study the whole ecosystem, we're going to need to go through a lot of stuff and be able to do it with reasonable throughput, not looking at it one cell at a time. But we also need to be able to look at the world from a microbial scale to see what matters to microbes, to see how it's affecting their interactions and their physiology. And so, you know, for this, my model is a wild microbe. It's the pinkberry that I'm showing you here. And so I really focus on this, and I like it as a model because it's a granular community. It's clear to me and my pea brain where the beginning of that community starts and where it maybe ends, where's its border, where it's interacting with other communities. And I'll show you kind of a number of other reasons why I think it's cool. So this sort of is an overview. Scott sort of gave you a touch point for this, but some of our toolkit that we use for studying wild microbes and sort of ranging. I used to have this on the bottom where it went from resolution to throughput, but the more I look at physiology, the more I don't think it belongs on this side. So I cut that out. But we have tools like microscopy, which you've all been learning to do in the lab, microscale biogeochemistry, which I'll talk about a little bit more. Metagenomics is something that I'm going to talk about today, and ways that we use this to look at everything from community structure to evolutionary dynamics and looking at how the strains have diversified. And then, of course, physiology, right? Studying isolates that we're able to bring into the lab, or monitoring just the ins and outs, the net production or consumption of the community can give us hints about its physiology. And we're going to do this in the context of pinkberries. But even if you didn't care about pinkberries, yesterday we heard a great talk from Beth Schenk about the tiny aggregates that occur within soils. And lest you think looking out in the ocean water that you might bring back from Eel Pond, or maybe you don't think it's very clear coming from Eel Pond, but from Stony Beach, you look at a bottle of clear water and it might look like, yeah, I'd expect there to be pelagic cells, you know, distributed like you'd expect them to be growing in a broth culture. But in fact, what you see is this incredible diversity of structural interactions that are happening, even in clear seawater at the micro scale. So you have particles that are sinking through the water and getting consumed by microbes. We talked about this a little bit in the field when we were talking about carbon production. So blooms of phytoplankton in the surface ocean that then die and sink to the bottom. And how does that control the flux of carbon in the global ocean?
And with all the technical issues that I'm having to deal with, I don't have the time to be going through all these emails. We need more fucking help.
Guys, someone needs to mute themselves because we don't want to hear about your email. Okay, so never a dull moment. You have the phycosphere here, right, around some of these diatoms that are growing and have specific microbes that grow around them, right? And then, you know, so this is an area studying this, obviously, it's invisible. It's really hard. It's dynamic and changing and water moves. This is an incredibly challenging system to study, but it's really, really globally important. Right. And so as you're thinking about why are we studying berries, you know, thinking about marine aggregates and how microbes come together in consortia, it can control cryptic fluxes of carbon, of sulfur, of nitrogen in the ocean, and at scales that defy our ability to, you know, put a sensor in the water or bring up a liter of seawater. And I've always been inspired by this idea, and I've just started to get a chance to do some of this work. In collaboration with my amazing friend and collaborator at UC Santa Barbara, Allison Santoro, who we've now started to try and do some of what I've been doing on berries on marine particles as they're sinking. And I think Braulio and I have discovered how very challenging this is compared to picking up a pink berry. This is a picture of some tracks that we made to catch sinking particles in a cruise 2 years ago in the Santa Barbara Basin. And sort of the difference in scale here, this pocket here is maybe, 2 inches across. So these are tiny compared to the size of the particles that we're working with. But the idea, the same principles hold. You've got microbes that are forming biofilms that are interacting in dense consortia with one another. And we're going to need some tools to study them in the wild. And so that's really where, coming back to my happy place, you can see why these might be an attractive, but still wild model to study because they're big, they're pink, they're easy to find. And amazingly to me, we have not yet harvested them to extinction despite the amount of time we've been studying them. So every year we can come back and they're still here. And so, just looking at the setting for folks who may be just joining us and didn't come with us to Sipawissat this week, this is the Sipawissat Salt Marsh. It's up in Falmouth, about 10 kilometers north of here. It's a salt marsh. It opens onto Buzzards Bay. And here in these little clearings of the Spartina eelgrass, we have these ponds. And beneath the surface of these ponds, we find these aggregates. So this is a remarkable picture that was taken by Scott Chmielewski with one of his really great macro zoom lenses. And you can see here that there are these sort of intricate little islands, almost looks like a brain, in a clear exopolymer matrix. Here's a zoom in of this even a little farther, and you can see these little islands. And I want you to stop for a second. And I want the folks who are here in the room, just turn the person next to you. What do you think these are, if we haven't talked about them yet? What are you seeing here? What do you think is happening in this photo? What do you think you're looking at? All right, here, and the scale is about 200 microns. I'm just going to give you a minute or two and talk to the person next to you. All right. What do you guys see? What do you think you're looking at here? Okay, George sees a biofilm. All right. Yes. And he's not seeing things. He doesn't just see them in his sleep. This is a biofilm. We've got lots of exopolysaccharide. That's the clear stuff there. All right, someone in the middle. Aggregates. Okay, yeah. So we've got these little islands here, these aggregates. Anyone have any ideas why they might be pink? What else have we seen that look like this week? Yeah, these are purple sulfur bacteria. Absolutely. So what you're seeing here are these dense aggregates. So these aren't individual cells, they're islands of cells. And they're clumping together in these little groups. Some of you might have seen this under the microscope when you're looking at some of your enrichments. Well, they'll grow in these little dense clusters of cells holding on to one another. And so when you look at this, there was a study that was done in '93 by a student in Alison Seitz and her classmate Peter Overman and Peter Nielsen and Jörg Overman, who was course faculty at the time. And what they did was they put some microsensors here into this aggregate, and they found that below the surface of the pink berry, you went about a millimeter in, all of the oxygen was gone. And they did some pigment analysis and looked at the cells, were able to determine that these were probably purple sulfur bacteria, just like you guessed. So we've got this unusual structure. It's not a mat, it's this sort of round ball, and it's got an internal compartment that's anoxic. And so this was a paper that I read when I was starting here, and it got me really excited thinking about these sort of from the microbial scale, right? The outside of this, it can sit in fully oxygenated water in the marsh. And we talked about while we were out there, you saw how dynamic the marsh is, right? We were there, we saw the tide go out as you were standing there, and then every day it comes in again and brings fresh oxygenated water. You've got oxygenic phototropes that are producing oxygen. And then you've got that really anoxic, organic rich mud down there. So it's a very dynamic environment where they're sitting. But these little buggers have been able to make a little core where they can live. And they have an anoxic zone that they've engineered for themselves to be able to survive through times in that dynamic environment when oxygen increases. So this is showing us, so Scott gave us a nice intro to this. This is looking at the percentage of 16S ribosomal RNA genes that we recovered from pink berries from a couple different years and sites. It's now, I suppose, to ancient history. So this was data that we generated. This was data from a past course student in 2007 who sent that to me. And then this was some data that we generated together. And what I want you to take away from this, so there's a fair bit of diversity in here, but I want you to take away these top two, which are that they're basically two species at the 16S level that make up most of the sequences that we get back from this. About 60% of the community based on 16S is only two organisms. And we'll talk about who they are in just a second. But thinking about that from a community complexity standpoint, that gives us the ability to do some cool stuff with this community because it's natural, it's wild. So if we look here at the rarefaction curve of different species, we've got a rare biosphere here. We've got a lot of stuff that comes and goes on the tail, but most of what we find here is just a handful of species, and they tend to be in common between different berries that we pick up, and they persist from year to year. So looking at who those top two species are, right? One of them you've ID'd by eye from 1,000 paces is the purple sulfur bacteria, right? It has these internal globules of elemental sulfur that it stores. Who can tell me something about purple sulfur bacteria? Where do they get their energy? Light. Who said that louder? Light. Yes, trust yourself. Okay, so they get their energy from light. They fix CO2 into their biomass. And in the enrichments that we were setting up, what was the electron donor that they were using? You guys remember? Sulfide. Yeah, good. So they oxidized sulfide to sulfate. Now, what was cool, that second species that we found that's most abundant and frequently occurring, now this was uncultured, so we don't have this organism in culture. We're just guessing what it does based on its relationship to other cultured organisms and its appearance, all right? The other species that we found was a putative sulfate-reducing bacteria. So you've grown these in the lab too, and these are organisms that are using sulfate as their terminal electron acceptor and reducing it back to form sulfide. Now, the way I've drawn this is suggestive, right? And it's suggesting that there might be a cycle that's happening at a very small scale within this aggregate, providing a local supply of sulfide to the phototroph in the community. And if we look at this is what we had in case the sand didn't work for you here. This is it drawn out in text, right? So sulfide here is in its reduced form, -2. And then going through the elemental sulfur that we have in these granules, all the way up to sulfate or sulfuric acid in the +6 oxidation state, right? So we've got a lot of potential redox activity that just looking at the 16S sequences suggested to us. Now, thinking about the electron donors that could be driving this, we have no idea, but I've drawn some arrows here saying, well, maybe it comes from the purple sulfur, or maybe it comes from the environment, who knows? That's the model we drew. But in case you thought that I was clever, and had come up with this. I didn't. This is a model that was drawn in an old course report where you can see the same exact cycle that he's suggesting. And this is work that was done by Udi Benin in 1997. And this was the first year in the course where they generated 16S sequences. And I read this report and I saw that he had reported that there was a Disulfopustis species and he had 16S sequences. They were only about 450 base pairs long. But when I was in the course, I emailed him and he said, I have no idea. I certainly haven't had that data. And he said, but if anyone has it, be Bruce Pastor at the Foresight. So I emailed Bruce Pastor. He said, oh, I'm going to have to dig up some very old drives and can find some kind of converters. But he found the data and he sent it to me. And it's really cool because it is still closely related to the same species that we find out there today. So we have something like now going on 30 years of data from these organisms showing that they've persisted through time in this environment, which is really cool. So the next step that then we took was to sequence the metagenome of these organisms and say, okay, how can we prove if this cycle is happening? And so we went through and we assembled, complete but fragmented genomes of these organisms and mined out the genes here, looking at the metabolic pathway in the purple sulfur to go from sulfide down to sulfate, and then from sulfate to sulfide in the sulfate reducer. And they have all the genes that you'd expect to be able to do this, which suggested, okay, maybe this is possible. But then we come to the question, do genomics match reality, right? Because this is just giving us the blueprint. It's not telling us that it's happening. It's not telling us that they're interacting. And so it's really, it's a hypothesis, right, that this could be happening. Of course, that's not the order that we did these experiments in, but it makes a good story to tell it that way. All right, so the first experiment that I did with my lab partner was that we put a bunch of berries in a bottle, so a very simple-minded experiment. And we thought, well, if sulfide is an important intermediate, maybe we'll see it get made. And so we put them in a bottle and we were very sad because they did nothing. On the y-axis here, we're measuring the sulfide concentration. We thought maybe they'd make sulfide and they didn't. And I was just so sad. They did nothing compared to the abiotic control. But when we fed them sulfide at about 1 millimolar, we found that if there was nothing in the bottle, they stayed flat. But the berries would chew down all that sulfide that we fed them within about 2 days. So they were a net sulfide sink in the environment. And in fact, trying to measure that sulfide concentration within the berries, we went in, we tried with microsensors, we still couldn't really measure it. And so we were basically saying, okay, well, sulfide's going in, but it's not coming out. So can we find any evidence for the other side of this cycle? And can we prove that this guy might actually be in there and be doing something? And so to do this, what we did was we built a phylogenetic tree of the 16S sequence for the pink berry. Ask me later and I'll tell you about this group of sulfate reducers. They're super cool. And a lot of these actually are symbionts from other sulfide oxidizing habitats, which I think there's a cool story there, but we still are very much still trying to scratch the surface of that. But we designed a fish probe, fluorescence probe, to try and go in and find this organism. And this was work that was done. We started it, and then it was finished by Verena Salmon, and then finished during my dissertation during the course. So, but here it is. So this is our sulfate reducing probe. And here we're visualizing in red the autofluorescence of the purple sulfur bacteria. So this is a cross section through those little tubules that you were looking at on some of the first slides. You can see here that the sulfate reducers are nestled right up against the purple sulfur bacteria. And this suggests why we may never actually have measured sulfide, because as soon as it gets produced, there are tons of these sulfide oxidizing phototrophs sitting around getting ready to suck it up, right? So this is in part, you can see why I kept talking about that your standing stocks are not necessarily the most sought after thing, right, in the environment. And so this idea of cryptic cycling, sometimes getting a view of where things are relative to one another can really suggest why that might be happening, right? And it has to do with the spatiotemporal scale of where they're occurring. Okay, so, but that still didn't tell us that that activity was really happening. It just suggested, okay, they've got the genes to do it. They're right on top of each other, so it makes sense. But it wasn't, we still didn't have a smoking gun to show that this was actually happening. And so the approach that we used was to use a stable isotope of sulfates. And normal sulfur is sulfur 32, and we used heavy sulfur. And we added this into the incubations with the idea that it would be reduced to sulfide, and then it might get accumulated within these intracellular granules so that we could then go after it and follow that label and see where it ended up. And this experiment was inspired by two women who've been incredibly important mentors and inspirations to me in my career, both of whom I met and got to know through this course. Nicole Dublier, who works on these gutless marine worms that have a very tightly coupled sulfur cycle within their skin between their sulfide oxidizing symbionts and a number of sulfate reducing bacteria. And Nicole did an experiment with radio-labeled sulfide and a silver needle that was very similar to this idea, to what I proposed. So I'd been reading her papers and I said, oh my God, could we try this? And the person who said, yeah, let's try that, you crazy, crazy grad student, was Victoria Orphan, who would go on to be an incredibly important mentor to me during my PhD, who was basically the enabler of me becoming a feral grad student. And said, yeah, let's do this. I've been interested in this for a while. And then I went on to do my postdoc with her group. And Victoria's lab, as Beth mentioned earlier, works on these anaerobic methane oxidizing consortia between bacteria and archaea. And so this sort of similar idea of looking at very small scale transfers that could be facilitating metabolism was very much in her wheelhouse. So we did these incubations. What's nice about the berries compared to seawater or other things is they're pretty easy to pick up and put in a bottle and remove from the complexity of the sediment and the seawater. So we can ask a focused question about this community by just plopping in a jar full of sterile water. And so we spiked our label in here. And then we took it to Caltech. This is in the basement of Caltech. This is the NanoSIMS instrument. It looks like as crazy a Rube Goldberg machine as it actually is. And like a racehorse, it's broken more than it ever actually runs. And so this, the long tube here is what's actually going to separate the ions into their different masses. So the way this works, you have your sample either on a slide here. We just took like a tissue section out of a pink berry. And then we blast it with a beam of primary ion beam of cesium ions onto a very focused spot size, about 50 nanometers in diameter, and then blow it apart as all of its secondary ions. So it comes apart as sulfur and nitrogen and carbon, and then you can focus those through that long tube in the SIMS to measure all of these, the abundance of these different ions at each spot. And as you raster it across the image, you can collect a heat map of the abundance of each of those isotopes. So that's what we did. And when I did it, I had no idea how incredibly painstaking and small scale it was. And I learned very quickly that there was no way we were going to analyze all 300 samples that I had brought from my incubations. We analyzed about 4 with a lot of SIMS time. So here is an overlay. This is showing you the carbon, nitrogen, and sulfur image that we're using to find sort of these ghostly halos of those coccoid purple sulfur bacterial cells. And then this is a ratio image showing you the heavy isotope to the light isotope of sulfur. And so what we see here is anywhere that's blue was the same abundance as our control with a 32S, a natural abundance, 32 sulfur. And then anywhere that's hot had taken up our label. So it was just chock full of label. And then we did a couple of variations of this where we added an inhibitor to the sulfate reducers. We could knock out this signal. We could wash it away if we dissolved away the elemental sulfur. And so we were pretty sure that this signal was evidence that we had this syntrophic transfer from one species to the other. So this is saying, okay, yeah, we've got this branch. We're pretty sure that this sulfur cycle is happening within there. But in no way do we understand what's happening in there, really. And so Braulio and Aaron are both now sort of working on the phase 2 of what I imagine was going to be chapter 2 of my thesis, and now that is chapters 1 through 5 of theirs. And so what they're working on broadly is looking at the carbon cycle in here. So how does carbon connect in? What is there a transfer of fixed carbon from the purple sulfur bacteria through to the sulfate reducer? What electron donors might this guy like to use? And can we understand its physiology better and how dependent it is on the other species? So they're doing this using a number of different cool approaches. So I'm just putting a little teaser up here so you can go talk to them later and ask them what they're up to, but they're doing some 13C bicarbonate stable isotope probing, some probing with 18O water, which will let us look at activity. And then combining this, not with NanoSIMS, because I discovered how very painful that is to do very slowly, but they're combining it with either DNA SIP, looking for the 13C ending up in DNA of labeled organisms as they reproduce, or in the proteins of these different organisms. And that's a collaboration between our group and Manuel Kleiner at NCSU. And so we're really excited about these next steps, and you should chat more with them. The other thing that Braulio has really been pioneering in my lab has been using fluorescence-based assays of activity. This is called BONCAT, or bioorthogonal non-canonical amino acid staining. You can see here, this is, they're lighting up bright green, and that fluorescence is coming from a synthetic amino acid that we've tricked them into eating. It looks a lot like methionine, and they accidentally put that in their proteins during an incubation. And then later, when we cut them up and fix them, we can conjugate it with an azide labeled dye, which is very specific copper click chemistry that just attaches that fluorophore onto the protein. So anything that synthesized new protein during this incubation will light up bright green. So I just, again, put this up here as a teaser to have you start thinking about ways that you can look at microbial activity that maybe don't require a NanoSIMS. So this lets you look at who is active during your incubation. And Braulio is using this to sort of probe some things both about spatial structure and also about which compounds stimulate the growth of different organisms in this aggregate. Okay, but zooming back out, One of the things that I love most about these aggregates, beyond the very small scale physiology, is that we can also take this and take a step back out. This is essentially a bacterial colony that grows in the wild, which is pretty awesome, right? And we've got lots of them. So we can sample, and from the first time I looked at this, one of the experiments I was most excited to do was say, well, are they all the same, right? A lot of times we grab 10 of them, grind them up, and hope they were more or less the same. But comparing the differences between each of these, we have individuals of communities that we can now sample in replicates. We find them in different ponds throughout the different marshes. So we have, the idea of this, and this was really inspired by reading Rachel's papers about hot springs and thinking about these islands where they might be occurring, and thinking about, is there a way we can look at this like a microbial Galapagos, and thinking about how they're diversifying in these different places. And then finally, we found them now first in Little Sipuisit and Great Sipuisit, which are about 1 kilometer apart, but now also in Penzance Point, which is right here near the MBL. And so you've got a spatial gradient that's really beautiful, that we can study how these things have been diversifying, we think, for at least 30 years. We have records of them happening over this spatial scale, and thinking about how this happens. So I have a lot more slides that I'm going to have time to get to today, but I'm going to give you a little flavor of what we've been doing here. So one of the things that's really beautiful about these berries is that they're big. They have lots of cells. There's almost no inorganic material in there. There's no sand. If you chew them, they aren't crunchy. There's cells all the way through. And so we can get a lot of DNA out of them. And what this has let us do is kind of use them as a metagenomics test kitchen to try some techniques, for example, that are only being done places where you can get lots of biomass. You try and do this in seawater, you'd be filtering for days. Sort of like what Trina mentioned in wastewater, this is a place where we can get a lot of biomass to try things to see what works. So this is me spooling DNA out of a classic Sandbrook extraction, where you get the DNA to precipitate, and instead of using a kit or a column, you just pull it out on a little shepherd's hook past your pipette and purify it away, because it's like a big DNA snotsicle. And this has been really cool, because for some of the new long read sequencing approaches, you need a lot of DNA. If you ask sequencing centers, they'll usually ask you for 10 micrograms of DNA. They're lying. They just hope that they, will be able to screw up a few times as they're doing it. We've done this from about a microgram of DNA, which is pretty minimal for PacBio, and we're now doing it with much smaller amounts, but still being able to test it with large quantities has really changed what we're able to do in looking at this. Now, the reason I started on this whole thing of trying to assemble more genomes was that the first genomes we assembled were really high coverage. We had between 200 and 1,000x coverage of the purple sulfur bacteria, which means we sampled the same position almost 1,000 times, but I still couldn't assemble it into one beautiful circular genome. And a lot of times when that happens, it's because you've got a lot of variation in your community, or microdiversity. So there are places where they aren't all exactly the same. And when you have Illumina sequencing, which is 100 to 150 base pair fragments, they can't figure out where to go. And so you just get shrapnel back. And that's pretty much what we were working with for the original genomes. So I started on this path of saying, okay, can we get better references? Because I really want to make this a model. And to do that, I need better genomes. And so, we started off a collaboration with PacBio to sequence the metagenome using long reads with this approach. And one of the things that was kind of an added bonus at the time was that the way that PacBio sequencing works is called single molecule sequencing. And as a base gets incorporated and is read out, there's a time factor here. So it's reading out one base at a time. And in some of them, you can see here, there's a pause after this A. And so this is actually due to the methylation of one of the bases. And so you're able to get information, not just about the identity of the base that's there, but about its epigenetic state. Has it been methylated or not? You can also see things about DNA damage, actually, from this type of sequencing too. And what we were thinking about from this point of view was that bacteria are able to identify their own DNA using methylation signals. And these are systems called restriction modification systems that are actually used for phage defense. So the bacteria modifies its own DNA so it can recognize itself, and then it has a corresponding enzyme that recognizes really specific sequences. And if it's unmethylated, it clips it up to defend it from phage. And the thought was, if bacteria can tell their own DNA from phage, maybe we can listen in on these signals to try and figure out how to put the genomes back together and better separate that complex mixture that you get out when you sequence a metagenome. And so what we did was we sort of set up a training data set to identify these sequence-specific motifs in the methylation data. And this is essentially where a methylation enzyme or a restriction enzyme would bind, and specifically recognize that. And then we use that to classify the context with the idea that if each organism had a different set of restriction enzymes and methylases, they'd have a different methylation fingerprint on their DNA. And this worked really well. I was really surprised at how well this worked. And what's cool about this is we can use these different signatures to cluster the contigs, and they both do things what we'd expect, right? They're coherent with other typical binning methods. But there's some cases where we never would have known that that should have been in the genome, except that it had this methylation fingerprint. And this is especially important in cases like phage, right? If there's a phage in the genome, a prophage that managed to get stuck in there, right? But it's on a real little fragment. If it has that methylation fingerprint, then it's snuck through and it's now become part of that organism. Or cases where you have horizontal gene transfer. This whole idea of the flexible genome, a lot of times are things that phylogenetically don't look like the rest of the organism. So if you try to use the similarity to that organism to bin them together, it doesn't work. And so you're missing the flexible genome when you try and look at it with that approach. But by looking at the methylation fingerprints, we could tell, yeah, this really only belongs to this critter because the other ones aren't methylated that way. And so we found some cool instances where we saw horizontal gene transfer of antibiotics and a number of other mobile elements and stuff like that. That was really cool. But most excitingly for me was I was able to close the genome. And this guy was a monster. And it took me the better part of a summer, like manually lining things up to finally, stitch these last bits together. And in part, that's because it's a really complicated bacterial genome. E. coli is about 4.6 megabases. This one is 7.9 megabases. It has transposases and CRISPRs and prophage. It's the whole 9 yards. And Braulio has been doing a really beautiful job going through and doing the metabolic annotation and thinking carefully about what pathways are there, what's complete, so that then we can complement it and look in more detail, sort of that level of analysis that we did with the sulfur cycle with carbon or with amino acids or with vitamin biosynthesis to see what other factors might be getting shared. The other thing that's really weird about this genome is that there are 606 transposons. And on mobile horizontal gene transfer week or mobile element week, you'll talk a little bit more about these, but these are essentially jumping genes that can insert and excise themselves from the genome. And most of the time we think about these as being potentially really harmful for an organism. If you've got something that can excise itself and insert itself in the genome, that's hazardous, right? What if it sets down in the middle of essential gene? This thing has up to 9.4% of its genome annotated in transposons. And compared to other closely cultured organisms, those only have like less than 1% that are in these repetitive elements. So this is just totally wild and unprecedented in terms of the amount of IS elements we see. What's neat from an evolutionary standpoint is the other place where we see that is in other high density bloom forming organisms like cyanobacteria and some other things. So it's not completely out there, but it's something we really do not understand from an evolutionary standpoint, why there, at least I don't understand why there are this many IS elements. Now, what you might be noticing on this assembly graph, so each point on this graph is a sequence read, and the edges between them are showing you an overlap between those reads. We've got these weird blebs. We call these assembly bubbles, and they're essentially evidence of two different paths that you could take in trying to stitch the fragments of DNA together. And these were evidence of structural variance that we have between organisms. So Scott was talking about sequence variance of an A changing to a T. This is a structural variant where we have something that's been inserted here relative to the other sequence. Now, what's really cool is we're now starting to do this. This is Hugo, really excited in the field, and we're starting to do this with individual aggregates using nanopore sequencing, which is another long read real-time sequencing molecule, or real-time sequencing technology. And what I took about 4 years and an entire summer of manual curation to do, Hugo's now having come off in the first round of uncurated assembly from his nanopore runs. So it's like the pace of acceleration in getting this in data quality is like off the charts. I mean, this project took probably the equivalent of 48 flow cells of PacBio data, which if we'd been sequencing it ourselves was about more than 50K worth of sequencing. And Hugo's now doing this with 8 berries on a single flow cell of nanopore that costs about $1,000 a piece. So the cost and the throughput and the quality of the data that we're getting out by being able to do it on one aggregate at a time has been just mind-blowing. And so what I wanted to show you was a better example of what those structural variants look like and what those transposon variations are. So we have at least, like, these are just 8 that I was able to fit on a screen, full closed genomes of the purple sulfur bacteria from different single aggregates. And what you can see here, this is a program called MOV that does whole genome alignment. So anywhere where it's yellow and tall, it's the same. These little dots probably are single nucleotide polymorphisms. And then here, this gap is showing you a place where this organism has something that none of the others do. It's got an insertion. And if you look at what that gene is, it's a transposon. And we see this all over the place. So there are very different arrangements of these transposons in the different strains. What's really cool, we finally closed the sulfate reducer genome, and we figured out why we were never able to do that before, even with our PacBio data, because you have to sequence one aggregate at a time, because the scale of rearrangements in the sulfate reducer is very, very different than the purple sulfur bacteria. So that the phototrophic organism is pretty clonal to pretty large blocks, and you get some of these transposon insertions and some cool flexible genes. The sulfate reducer is just off the hook. About every 10 kb, you have a massive block of something that comes in and moves out. And it's just very clear why its assembly was always just shrapnel, because they seem to be much more clonal within an aggregate than between aggregates. And this is all data that, like, Hugo has sent me in the last 10 days. So stay tuned. But it's really cool in thinking about how these strains have evolved and being able to really look at the flexible genome, not guess where it might be, or culture the organisms out of the environment and then sequence them. We can go and grab them in the wild and see what they're doing. And what's fun about that is we're now using the spatial variation that I was excited about, right? And we've got data from about 300 berries across 3 marshes in a mixture of different sequencing technologies. Some of it is short read Illumina data that we started off doing first, and now we've got about 100 with the long read data that we're working on. And from the preliminary analysis, this was a collaboration with a great postdoc named Gabriel Leventhal, who was at MIT. And looking at this, we can see that between these different sites, we see different community structures. So they all have the purple sulfur and a sulfate reducer, but we do see some variation in those other community members and the carbon degraders and other species that are there sort of in accessory. And we also see population structure. So this is a whole genome phylogeny of the purple sulfur and the sulfate reducer. And we see that those populations appear to be geographically structured. Now, this was all done from short read Illumina data with just single nucleotide polymorphisms. So the next version of this tree, I think, is going to be much more interesting and illuminating when we look at it in the context of the complete whole genomes that we're getting back. But what's cool is when you look at what genes are diversified in this organism, and I think this is a nice lead into Rachel's stuff next week, looking at viruses and other things. I really did not want to study viruses. I'm afraid of them. I don't understand if they're alive. I don't understand how to organize their diversity. I really was not going to study them, but all of the genes that are most clearly diversified between these different populations are viral genes. So when you look at this colony, right, it's a sitting duck. You've got something like 10 to the 10 or 10 to the 11 cells that are all closely related to one another. And if a phage came in there and started replicating, it could spread like wildfire and kill everyone, right? And this is essentially the idea of social distancing. These are not well social distanced at all, right? They are not better apart. They are very closely together. And so what we're finding, the more we look at these genomes, is they do not look like normal cultured creatures. They do not look like normal cultured pelagic bacteria. They are weird. They are full of all of these different viral defense strategies. So they have things to try and detect the threat and look for self versus non-self. And that's really the methylation systems I was talking about. Things to search and destroy or find those phage and go after and clip them up. So they have CRISPR-Cas abundantly throughout them, and then they also have all these restriction enzymes. And then we also see this idea of kill yourself to save your kin. And this is the idea of abortive infection or toxin antitoxins. So if a cell detects that it's been infected, there are a number of different mechanisms in cultured organisms that are described where it can sense that it's infected and then its defenses are not working, and then either kill itself by apoptosis or slow its growth enough to prevent a phage burst. And we're seeing a lot of potentially novel mechanisms of what I'm guessing are doing that. But they're really weird, they're pretty novel, and a lot of them are all associated with multicellular bacteria, biofilms, and these types of habitats that aren't very well studied from organisms that are adapted to survive and persist like this in the environment, which is kind of a weird thing to do. And then there's this idea, if you're going to be using this strategy, kill yourself and save your kin, you better know when to pull the trigger. And so we're also seeing some cool methods and adaptations to figure out when is that happening and to decide where's the threshold where you cut it off. And with that, I'm going to skip over some of this other stuff that I have here at the end and just talk about, so we're doing lots of stuff with phage. We're going to go out and filter a number of the berry ponds and look at how the pelagic viruses in those ponds might differ and whether that changes the strain evolution. But beyond phage, we see other predators too. So one of the metagenome assembled genomes we've pulled out is a novel genus within the Microbibrianaceae. And this is a cool, I just found this morning, George, a cool connection between our labs. So the described and cultured organisms in the Microbibrianaceae are what we'd call a vampire bacteria. These are predators, bacterial predators that come up and in a pretty specific association, find a host, and then they attach to the outside and they suck out their guts. And these, the described cultured organisms of these, you can see here, this is some work that George came out of this paper that George was on from 2007, where they grew Pseudomonas aeruginosa, and then they grew Microbial aeruginosaurus, and find that it just decimates that biofilm. And so thinking about this, there's a lot of things. So the one we found is not within this genus, Microbibrio. It's a distant relative. There are no other cultured organisms like it. But my student, Tori Jones, who was an undergrad extraordinaire working on a remote COVID research project this winter, has been analyzing this genome. And this genome is super weird. It's only about a megabase and a half. It's missing all these amino acid biosynthetic pathways, and it very much resembles what we know from cultured Microbibrio that are missing many of the pathways that you would need to be free and independent. And so that's Tori's master's project is going to be going after. So these are pictures from the cultured distant relative. We're going to go try and find it. So she's designed a fish probe to go and look for it. And we're going to be hunting for that in the environment, trying to see if we can both see both viral predation and also this type of bacterial predation. So that's pretty much what I've got, thinking about these interactions on this scale to the landscape scale, looking at these sort of buried islands and trying to think about how does this type of really tight interaction, we know this happens in other ecosystems. It's just not as easy to pick them up and find them and study them year after year, to have your fish probes work for the same species when you come back to it next year, right? That's a convenience that this is really letting us get at that kind of bridge of mechanistic questions that I really love, right? I love being able to go out and get dirty in the marsh, but I also love being able to ask questions that have to do with metabolism and chemistry and the physiology of these organisms, and then put that in their evolutionary context. How do they come into being, and how are they interacting with one another and their environment to where we have them now? So with that, I'll stop and just say, come join us. We're looking for, always looking for good folks, particularly postdocs. So if you are here in the room or remotely, come talk to me. We definitely are looking for folks to come and be more berry fans. So with that, I'll stop and take any questions you guys might have.
Thank you, Lizzy. We're going to move on to the Q&A section now. The virtual audience is free to submit their questions via the Q&A function. And Lizzy, if you take questions from the audience, could you just repeat the questions for our virtual audience before answering them?
Yeah, absolutely.
Thank you.
Yeah, or I can just repeat them if folks are shy. Yes, Dallas.
So here in the transpose on, you think that rearrange it more of a selective that determine how much they rearrange, or could it have some way of separating for like impact the aggregate versus outside the aggregate, and so then they would do, rearrange it, determining where they are.
Yeah, so Dallas's question had to do with the transposons that we looked at and whether they were being selected for a particular random rearrangement or whether they could be actually a response to some environmental trigger, whether you're inside or outside the aggregate or something like that. Is that, or who you're with, or something about your context that the microbe might be sensing? That's a really good question. The short answer is I have no idea. There are a number of different thoughts about why there might be so many transposons, and some of them have to do with the idea that there could be relaxed selection. So the cases where we know of transposons proliferating like this are often inside intracellular symbionts of insects and early in the symbiosis. So you have this case where you've got a reduced population size and relaxed selection. And so it kind of doesn't matter if you're sloppy. And so they're allowed to make mistakes and they can persist. But that really doesn't, to me, make sense in this system. So I don't have a good explanation for that. Some thought is that maybe it's some kind of phage decoy. So if it's coming in, maybe it could be distracting something and being able to come out. The other big thing for having a lot of transposons is they also are a catalyst for homologous recombination. And so even if there wasn't a selection for that transposon, by both breaking up DNA and resplicing it, and then also just being a repetitive element, it gives more places where you could have an overlap and have that homologous recombination happen. And we see a ton of homologous recombination in the genomes for these things. So that's an idea. I also need friends who know more about this type of population biology. Rachel, please help me to help us think through that. But the other thing that you suggested about some type of sensory or functional mechanism is also a really exciting idea. So there are a number of bugs, and this is more well described in the clinical literature, where you have phase switching between, for example, being a biofilm former or not being a biofilm former. And that switch can happen by the deliberate excision and insertion of a transposable element near the promoter region of some important functional gene. And so we could, exactly like you suggested, be catching it in some phase switching state between those different organisms. And that's described not just in pathogens, but also in Alteramonas macloidii, which is a particle-associated copiotroph. So there's definitely reason to think that there could be a functional role there. And that's something that we're excited to look at in some of Braulio's data. He's going to be doing some metatranscriptomes this summer, and we're excited to look and see if we see them being transcribed, or like you said, are they just vestiges? Did it get in there and now it's been selected for and it's just persisting like that? Or do we see a lot of activity going on in moving them around and changing that? Yeah, Scott.
How do berries grow?
How do berries grow?
I mean, maybe
Yeah.
that's one explanation for sweeps of
Yeah.
versus if it's the clonal population.
Yeah, so I don't have a good, we don't know. The short answer is I have no idea. We've seen single cells and small aggregates of them in the, and by we, I mean Verena Salmon in her course report from 2011 has filtered berry pond water and found small aggregates that are the purple sulfur bacteria, sometimes in association with one or two sulfate reducing cells. So they could form in the water column and then sink down. Those could also be artifacts that sloughed off the outside of the giant carpet of them down below when we walk around on them or something. So I don't really know. We can't get them to grow in the lab. We've grown a relative of the purple sulfur bacteria in the lab, but recently found out that 97% 16S similarity is not enough. And its genome is half the size of the wild one. So that was a perhaps needed lesson in how different. So we now have the closest cultured relative, also from Superwicet, also from a berry, but that's not the dominant form in the environment. The 1998 crew had a theory that they were muscle poop. I don't think so, but you know, mussels could eat them. So we definitely have seen them in our, they're not totally an island. We've seen fly larvae hatch out of them. And we had, when we were out here in 2019, we had all these little red worms that were some type of black fly larva that was growing inside them. And there's actually some cool isotope data from older papers in Great Sipawisit that showed that a lot of the macroorganisms out there, the consumers out there, have an isotope signature of about -20 per mil in the delta 13C. And that's not consistent with Spartina as a major source, but it is consistent with purple sulfur bacteria and with other sort of autotrophic bacterial sulfate reducers like Vegetoa. So they could be a major food source out there also, but we really don't know. The closest thing I have to a theory about how they form comes from looking at upflow sludge reactors. Yeah, I know Tina's dancing. So upflow sludge reactors are these settling type reactors where you get wastewater coming in from the bottom, and then the hydrology of the system makes them form these really dense, stable, sturdy flocks that then sink back down. And so we think that some of the selection for the berries is very similar to the selection for the flocks, in that they want to stay in these sulfidic organic rich ponds. They don't want to get flushed out to sea. They don't, you saw those tidal channels with the fresh oxic water and the sand, they were much less sulfidic. So they want to stay in those muddy, mucky berry pools, and they don't want to get washed out to sea. So we think that settling might be part of the effect of how they get selected for and formed. Yeah, Emily. Size does matter. Yeah. Good question. We should try.
They reform in a few days. And here, if you grind up the berries from different locations, would they intermix?
That's a great question. Okay. So the question was, if you grind up berries, this is Joe Bellino at the Ecosystem Center. Thank you, Joe. So the student who he's talking about is Rachel, and I'm blanking on her last name, but she's great. She's at USC. And so she did this experiment where she ground them up, let them reform, and saw that they reformed. Now, they're very sticky. So whether they reform in the same way that they were before, we haven't looked at the microstructure level, but they definitely are sticky and goopy. And so they will come back together. Whether they form the same type of islands and tubules, and whether all of the same species survive during that reformation process, we don't really know, but it's a really good question. And I think the second question of, if you combine berries from different sites, what would happen, is so cool. So my theory is that they would not like each other. And I think you would see competitive exclusion of different types. And I think this has to do with, they have particular weaponry on the outside of them. One particular type that I think would be the most problematic is called the type 6 secretion system. And this is like a nano harpoon that they have that will detect whether it's near a kin cell or a non-kin cell. And if it doesn't have the right type of response to the kin, it will kill it with a harpoon. And so we know that they have type 6 secretion cells. We haven't studied the mechanism of any of that, but my expectation would be that they're quite picky about who they would associate with. But that's definitely the direction that we want to go in looking at these strain level differences. How does it control who you're interacting with? Or is it just random, right? Do they not care? Maybe that has some other function that we're not cued in on, but that's a really, really good question. Thank you. Yeah, in the back, Bob. Are there evidence that they're hotspots of? Are there evidence? Yeah, so Bob's question was, is there evidence statistically that these are hotspots of transposon insertion? I'd say absolutely. Do you mean hotspots within the genome where that happens? Yes, yeah, there are very specific sites where it does seem to happen. Some of them, we don't have enough evidence from enough different things to say, but we definitely see often near these recombinase sites. So you'll see like a tyrosine recombinase, and then you'll see a string of transposons that have gotten plopped in there. Yes, both. You will see both. Often you'll see duplicated ones, and then you will see some diverse ones from different systems, but you definitely see hotspots in the genome, and that's consistent with what people have seen in other more sort of comparative genomics studies from isolates, but we see a similar thing, but we need to get a better handle on it. We just really, I think just, within the past 2 weeks have the data we can confidently say that from, because before we were analyzing that on mixed samples, just looking at the read alignments and the population diversity within one sample. But now that we have pretty much clonal genomes from separate aggregates, we can really start to answer that in a more satisfying way. But yeah, I think that that's definitely something that's happening.
There's a question that came in really early. I'm gonna ask that one. This is from Steve Lindner.
Hi, Steve.
What are the common members of the berries in the Galapagos?
Yes.
Could this be another predator?
Oh, so Steve's question was, there's a common member in the pink berries is a Cytophaga organism, and Steve's asking if it could be a predator. It's an interesting question. So Cytophaga, are you guys doing Cytophaga enrichments? Yeah, has anyone seen them yet under the microscope? What do they look like? Someone give me a hand motion. Oh, they're long, right? For those who are on camera, they're long. And so they're long filamentous bugs within the Bacteroidetes, and they are often facultative anaerobes, and we think of the cultured ones as being high molecular weight organic carbon degraders. So we see them, Braulio and I have seen them. Sometimes we call them the Cytophaga party, because we'll see them in the activity data and they'll just be like lighting up like a Christmas tree. And we'll see a whole bunch of them in one region, like a pile of snakes. They're very visually distinctive, even though we haven't looked at them with a fish probe. It's just very clear what they are. And so Steve, could they be a predator? To be a predator, they'd have to be going after the purple sulfur bacteria while it was alive. And they could. I don't know, maybe they are not symbionts. Maybe they are predators. We've always thought about them as degrading that polysaccharide or EPS goop once it had been secreted. And I think that the Cytophaga parties that we see sometimes are near recently lysed cells, potentially. And so we definitely see hotspots to their activity, whether they wait for them to die a natural death or whether they go and make sure that that happens to feed the family. I'm not sure, but that's a really cool idea. I like it.
This is kind of similar to that last question. So there's ponds in these. It seems like kind of a bait, like predator and other things. Like, do you have any idea of how they're able to persist? It seems like so much food there.
Yeah, good question. So some of the defenses that we were talking about, so here's one of them. This is a slide I skipped, so thank you for giving me a chance to go back to it. So this is a CRISPR-Cas array. So the Cas genes are up here. This is a CRISPR array. For those of you who maybe haven't looked at CRISPRs before, they're essentially added on like a cassette tape. So it will sample, you don't even know what a cassette tape is. Like tracks on a playlist. And so each one of these is a repeat that's specific to the CRISPR. And then the little white gaps in here are the bits of foreign DNA that it's sampled, that it's seen. So they're like infections past, and it just grabs like a 20 nucleotide snippet of that and records it in this array. And so what we're seeing here is that most of the older ones in this population that I'm showing, this is a reference genome up top. These are all sequence reads down below. So the older CRISPRs that are added from this side going that way are shared. But you can see here 3 sequences that came from an organism that was missing those or had a different CRISPR spacer at the end. So they definitely have lots of defenses for things that are coming in. They've got other crazy defenses that we can talk about that diversify themselves. So if you think about the arms race, like looking at like sort of a red queen hypothesis where they're constantly evolving to escape their phage. We see some specific types of diversification, retroelement guided diversification that are constantly doing a site-specific mutagenesis of some target genes that we think are interacting and involved with phage defense. And this is some work that is a really fun collaboration with Blair Paul, who used to be a postdoc at UC Santa Barbara and is now a scientist here at the MBL. And we're working on some of these methods of viral defense and diversification, and trying to figure out what they're doing. But they definitely have lots of weird defense stuff. So yeah, it's a good question. Yes. She was sharing that they dressed up as berries in 2012 for the parade. Mm. Why are the berries squishy? That's a very good question. And they're really tough too. So compared to dealing with other types of aggregates or flocks where you have to handle them very gently, these ones you can put in dish and swirl them and they really do not come apart. My guess is the ubiquitous exopolymeric substance. So we've thought about everything from, you know, carbohydrates to actually DNA as a potential polymer that could be, which would wreak havoc on why we have such a hard time isolating high molecular weight DNA from these aggregates, if in fact extracellular DNA is sticking them together. But that was actually
Sorry, everyone. We're not sure what happened there. Unless we can get her computer back.
the purple sulfur bacteria. Oh, did that mean that we got cut off? Are we back?
You're back.
Okay, cool, we're back. Sorry, my laptop died because I didn't plug it in. Good timing. So at night, the purple sulfur bacteria don't fix carbon. They use their intracellular sulfur stores or stock up on sulfide that's coming out of the sediment. And they do a cool type of maintenance metabolism where during the day, as they fixed carbon, they have glycogen granules. So these are a storage compound of essentially it's a glucose polymer that they store. And at night, they take that polymer and they ferment it. And so, but if you normally fermented glucose, you would get fermentation products like acetate, hydrogen, you know, other sort of organic acids. But that sort of is terrible if you're a phototroph and you worked really hard to fix that carbon because that carbon was expensive when you fixed it. So the purple sulfur bacteria do a cool thing where they ferment it not to acetate or excreted acids, but to polyhydroxyalkanates, like polyhydroxyvalerate and polyhydroxybutyrate, which is actually the ingredients to sustainable bioplastics, which is kind of neat. So these things are chock full of little bioplastics granules. And when they do that fermentation, they're able to save, they don't make quite as much ATP, but they save about 4 of the 6 glucose carbons. And then during the day, they can convert them back to glycogen. So they're able to do that for maintenance ATP respiration at night, but then they've got this cool polyhydroxybutyrate production inside their cells. We know that the environment there gets a lot more sulfidic at night when the oxygenic phototrophs get knocked down. So we see the sulfide in the marsh move from sort of in the sediment to all the way up in the water column, maybe 4 centimeters up in the water column will be sulfidic. So, all right, it's cutting me off here. That's the cane. But yeah, we don't know a lot about how the physiology is changing, but that's something that Braulio is looking at this summer. And so he's going to do a transcriptome over a 48-hour time course. So when he's looking really sleep deprived later in the summer, that's what he's working on, is trying to figure out what they're doing at night and how the metabolites might be changing, because that's a really cool sort of experimental setup because they've got that diel variation built in. Any other questions? Ah, James Hendrickson was here. Oh, hi, James. Sorry. Trina, yeah.
Do you have an idea what the photosynthate that they are providing to the?
No. The question from Trina was, do we know what the photosynthate might be? that they're providing to the SRB. My guess is it's some degradation product of the exopolysaccharide. We actually think it probably isn't direct. So I don't think that the sulfate reducing bacteria can directly use that secreted polymer, probably. I mean, maybe there is something direct. So one of the theories, the closest relative of the sulfate reducer is disulfophysis glycolicus, which is a glycolate oxidizer. And glycolate is a compatible solute that's used for salt tolerance. And so that's something we're going to look for to see if, so we're doing some measurements this summer in collaboration with Shady Amin at NYU Abu Dhabi, where we're going to measure the exometabolites that come off the berry. So we're going to put them in some blank seawater and then see what comes off and measure that. So glycolate is definitely something we're looking for to see if that's there.
But there you have the gross beer problem.
The gross beer problem. Yeah. Yeah, exactly. Yeah. So is it going to be there? So one of the things we're going to try is inhibiting the sulfate reducer. So we're going to do it with and without. So there's a specific inhibitor for sodium molybdate, or a specific inhibitor of sulfate reduction called sodium molybdate, which is a sulfate analog. And so the theory is if we knock down the sulfate reducers, whatever it was that they were eating, we would hope might build up. Maybe it just poisons everything and we don't see something sensible, but that's the hope, is that we could try and see some of those hidden pools. But yeah, that is the problem. So what we're trying to do is probe the berries with a number of sort of candidate compounds like glycolate or acetate or some short oligopeptides, because it's got a lot of peptide transporters, and then look how their activity changes with the 18O water proteomics experiment. So see, are they more active if we give them glycolate? Are they less active if we give them lactate? That sort of thing, to see what they might like, sort of start narrowing down that field. But that's what I'd really love to know, right? That would help us close the loop and figure out how they're all fitting together. But we've been struggling. It's a hard question. Any other questions? I'll also be around all summer, so you can keep asking. Yeah. Yeah, so Nyla's question was, from looking at the genome of the sulfate reducing bacteria, do we have hints about the carbon source? And the short answer is yes and no. So one of the challenges, so we should be able to do that, but part of the problem comes with the specificity of the annotations. So if I believed all the annotations, then I could tell you perfectly, but particularly for transporters and things like that, a lot of times we can get as close as, it's probably a 2-carbon compound that's a carboxylic acid, or it's probably a 4-carbon compound trap transporter, or it might be an oligopeptide. So we can see things like that, and we're definitely want to look more at that, but we haven't looked at it in as much detail as we should. We definitely see some, so you can look for transporters. You can also look for dehydrogenases, so things that are going to be ripping it apart and pulling its electrons off, right? And again, how much you believe exactly some of the specificity of those, many of them are kind of distantly related, so it's a little bit hard to tell, but yeah, we're definitely getting clues from that, and that's something we're going to look at more. It's a great question. All right, with that, I'm going to let you go to lunch. Thank you folks so much. It's been really fun.
Thanks, Lizzy, and thanks everyone for attending. We hope you'll consider registering for the other lectures in our series. And just a reminder to register for Global Pink Bang